This is a stark reality, but at the same time, it also points us to the critical importance of innovative approaches, like and the promise of precision oncology seeks to tailor treatment to individual patients using data-driven insights. I'm particularly excited about the potential in precision oncology, big data, data science in biomedical and health research, because it will help us Africans to overcome the deficit of economic resources and also potentially transform health outcomes for many cancer patients and hopefully prevent cancer deaths as well. So today we are thrilled to dive into the topic from data to discovery, computational systems medicine for precision oncology. It sounds like a big topic, but our experts, we have the right experts to break it down for us. Our distinguished speaker is Professor Tero Aiittokallio. Did I get that right? Right. He's a pioneer in the field of precision oncology and health data systems. So we will introduce our speaker, but just to say that he has built a very grand career in this field, and his experience spans biostatistics, machine learning, and artificial machine learning. Okay, so machine learning, AI, and all that. So he's coming to us from the Oslo Center for Biostatistics, and he's also the Center for Epidemiology, Center for Biostatistics and Epidemiology in Oslo, at the University of Oslo. His work has been instrumental in advancing functional precision medicine, helping to identify synergistic therapies that could make a difference in patient care. So we'll invite him to share his insights with us in a 30-minute presentation. But before then, we will take the opportunity to showcase some of the research that is being conducted at the Center for Research in Applied Biology here in UNE. So that is how basically our session is going to go. Then we'll break for a photo session, and after the photo session, we'll have a closed collaborative discussion with our heads of department, staff of the research center, and so on. So, ladies and gentlemen, without wasting much time, I would like to kickstart our session by introducing our dean, the dean for the School of Sciences, under whose auspices we are having this engagement. So our dean is in the person of, he needs no introduction. He's in the person of Professor Samuel Fosu JC. He's a seasoned microbiologist, and he was instrumental in the establishment of the Center for Research in Applied Biology. So, ladies and gentlemen, shall we please put our hands together? My name is Emmanuel Timidonko. Shall we please put our hands together for Professor Samuel Fosu JC? Shall we please put our hands together?
Thank you very much, Dr. Emmanuel Timidonko. On behalf of the Vice Chancellor of this great university, Professor Efesa Saribediakon, I would want to welcome you all to this special program, and especially our guest, Professor Tero Aiittokallio, because of whom we are here. Let me also take this opportunity to also commend CeRAB, Center for Research in Applied Biology, under the able leadership of Dr. Emmanuel Timidonko and his team of hard-working officers for the wonderful work that you are doing to increase the international visibility of this university, and to a large extent, School of Sciences. I want to believe that such visits are important because as we expose ourselves in the international arena, we get to learn from seasoned professors who are doing so well in the area of work. I have no doubt that Professor Aiittokallio, worth of experience stemming from mathematics, statistics, into system biology, and finally into cancer, would have a lot to say. It's my prayer and hope that all of us, coming from the nook of cranny of research, far and wide from this university, we get to learn from the depth of his experience. On this note, I want to thank you for coming and hope that you'll be with us to the end of this presentation. Thank you very much.
Thank you very much, Prof. So we are going to go right. So also, as we welcome Prof, I also want to take this opportunity to welcome all our heads of department, all our professors from the School of Science and from the university at large. We are deeply honored by your presence. So we'll go straight into our first presentation. Before then, I would like to read a short bio to highlight the profile of Professor Tero. So Professor Tero Aiittokallio received his PhD in applied mathematics from the University of Turku in 2001 under the supervision of Professor Matt Geilenbeck. And he then did his postdoctoral training in the Systems Biology Lab at the Institut Pasteur from 2006 to 2007 with Dr. Benno Schwekowski. In 2007, Dr. Aiittokallio launched his independent career as a principal investigator at the Turku Biomathematics Research Group, where he received a 5-year appointment as an Academy of Finland Research Fellow. Tero Aiittokallio joined FIMM as EMBL group leader in the fall of 2011 and was selected as Professor of Statistics and Applied Mathematics in 2015 at the University of Turku. So if you look at his background, you see how he's transitioned from a purely mathematics background into precision health and precision oncology. So in 2019, he started as group leader at the Institute for Cancer Research, Oslo University Hospital, and professor at Oslo Center for Biostatics and Epidemiology, also in the University of Oslo, Norway. His group, the group that he leads, has expertise in integrating multi-omics profiling and clinical information from cancer patients using mathematical and statistical approaches such as machine learning and network modeling. The medical aim of his group is to optimize treatment outcomes for patients using maximally predictive models and minimal biomarker signatures that enable real-time and cost-effective routine diagnostics and prognostics. So ladies and gentlemen, shall we please put our hands together once again for Professor Tero Aiittokallio. Obviously, he's achieved a lot. But before Prof. Aiittokallio delivers his lecture, we'll have a first lecture presentation on some of the foundational work that we are doing at the Center for Research in Applied Biology. I know many of you have questions about what we do and all of that. So to that, I will introduce Alfred Efriye. I welcome Alfred Efriye. Without a long introduction, Alfred is one of us. We have many more opportunities to introduce ourselves. So Alfred, please come to the podium and as you deliver our first presentation. Shall we please put our hands together for Alfred? He's a senior research assistant at the Center for Research in Applied Biology. He's presenting on dual inhibition of the SH2 domain and P110 beta catalytic subunit. Dual inhibition of the SH2 domain and the P110 beta catalytic domain promotes cell death in triple negative breast cancer. Shall we please put our hands together for our presenter?
Thank you very much, Dr. Time. Without further much ado, we will go straight into the presentation since we are behind time already. All right, so dual inhibition of SH2 domain and P110 beta catalytic subunit promotes cell death in TNBC, that's triple negative breast cancer. So the presentation outline follows the usual. Before I start, I would like to explain to you that this is a proof of concept of a bigger project that we are working on at CeRAB, which involves in vitro studies and in vivo studies before clinical trials, if possible. So this is just a proof of concept, which takes out a part of the computational aspect, that's bioinformatics of the project, and that's what I'll be presenting to you today. So the presentation follows the usual outline of introduction, aims, methods that will be in silico, purely in silico analysis, and results and discussion, including conclusion and your input and subsequent studies. So TNB. Hello? Yeah. So TNBC is a subtype of breast cancer. We all know breast cancers. There are a lot of breast cancers that we are known of. So TNBC is a subtype of breast cancer, which typically lacks the usual 3 receptors that are usually found on all subtypes of breast cancers. That's the estrogen receptor and progesterone receptor, which are intracellular receptors, all right? These are not membrane-bound receptors. Then we have the HER2 receptor, human epidermal growth factor receptor 2, which is a membrane-bound receptor. So usually in TNBCs, these receptors are absent, and usual breast cancer treatments often target these receptors. So the absence of these receptors makes it difficult for usual drugs to treat this subtype of breast cancer, all right? So this particular project or study will be looking at exploring other ways, okay? We are trying to find other ways where we can treat TNBC by bypassing these 3 receptors. Okay, so the biological features of TNBCs often show gene expression, which are similar to the basal epithelial cells in the breast, which are usually involved in structural function of the breast, okay? That includes pushing out of milk. The similarities often lies in the absence of these 3 receptors in basal epithelial cells. So that's where the similarities lie. However, TNBCs often show EMT markers, that's epithelial to mesenchymal transition markers, okay, such as the N-cadherin. High levels of N-cadherin in TNBC or screening tests often show metastatic TNBCs, okay? So where Aiittokallio, that's an epithelial cadherin, which is involved in adhesion, okay? Adhesion of cells, transmit or transfer into N-cadherin, that's neural cadherin. So you are transferring from cells that are static or adhesive cells to cells that are mobile or motile, okay? So they also show high proliferative rates, and they show aberrant mutations in TP53 gene, which is the guardian of the genome. That's the security chair or whatever of the genome, okay? That's the TP53 gene, and the BRCA12 gene. It also shows high immunosuppressive tumor microenvironment where PD-L1 cells found on PD-L1 ligand, that's a ligand program def ligand 1, found on cancer cells often target PD-1 receptors on normal cells, suppress them, on T cells, sorry, that's immune cells, suppress them, then evade immune clearance, okay? So TNBCs often show high immunosuppressive tumor microenvironment. Breast cancer, usually TNBC often is made up of 15% of breast cancers are usually triple negative, all right? So, and it's very predominant or very prevalent in Black populations, and this is why there is a need for us to do this project in our setting. Geographically, West Africa, as you can see there, Ghana shows a high incidence of TNBC compared to other places in Africa. And this further confirms the reason why we need to do this project. So emerging treatments in TNBC include immunotherapy, where PD-1, PD-L1 inhibitors such as atezolizumab, which is a monotherapy, often targets this PD-L1 found on cancer cells, that's TNBC cells. They are PD-L1 inhibitors, basically, that's the meaning. And we have PARP inhibitors, that's an enzyme which aids in assuming you have a mutation, the mutation needs to be corrected. This PARP enzyme is responsible for recruiting mechanisms or factors that aid in genetic repair, DNA repair, basically. So these PARP inhibitors, cancer cells usually hijack this particular enzyme to repair cuts caused by treatments that are being used. So PARP inhibitors often target PARP enzymes in order to stop this DNA repair. Then we have ADCs, antibody drug conjugates, such as sacituzumab govitecan. So this particular drug was used in this particular study as a standard. And it's an intravenous infusion drug, which was used as a standard control in this particular study. There's also multi-targeted therapy, which is another standard which was used, and there is phytotherapy. And that's where we'll be basing our work on. So the aims and objectives of the study include exploring the dual inhibition of CH2 domain and PI3K P100 beta catalytic subunit of TNBC via bioinformatic tools or computational modeling. So the objectives will include screening bioactive compounds derived from Nigella sativa. Nigella sativa is what we commonly refer to as black seeds. So compounds retrieved from Nigella sativa will be screened against the selected targets. Then we will evaluate the binding affinities via molecular docking. Then visualize the interaction at the binding pocket using Discovery Studio. So the targeted oncogenic pathways, the first one being the SH2 domain, which is a domain part of the STAT3 molecule. That's an intracellular signaling factor, which aids in the dimerization of STAT3 molecule. Okay, so it aids in the dimerization of the STAT3 molecule, which then translocates into the nucleus and undergoes, binds to the DNA inside the nucleus, leading to malignant activities such as anti-apoptosis, proliferation, metastasis, and drug resistance in cancer cells. So what we'll be doing is we'll be targeting this SH2 domain, which aids in the dimerization. Once we are able to inhibit or block this dimerization, then cancer cells will not be able to cause these effects. So that's the concept behind this target. Then the next one, that's the P100 beta catalytic subunit, which is a subunit part of the PI3K beta, which forms a dimer with P85. P85 is another molecule which is involved in the PI3K MKT, AKT mTOR pathway. So once we are able to stop this P110 catalytic subunit, there wouldn't be the existence of PI3K beta factor. So it's more like we are trying to block the PI3K beta and its function. So why the need for these two targets? We could have just done one, right? Yeah, so the need, as it's seen here, targeting both PI3K beta and the STAT3 SH2 domain offers a block to a common escape mechanism, where studies have shown that blocking one leads to the hyperactivation of one. So this is why we decided to block the two, to target these two, all right, to block that common escape mechanism. Additionally, PI3K beta also reduces tumor cell survival, while STAT3 inhibition enhances anti-tumor activity. So this promotes a stronger immune surveillance whereby cancer cells are not able to evade the immune system. Then the last one, P10 deficient TMBC. P10 is found in the PI3K AKT-mTOR pathway, which actually serves as a control, okay? It's an on and off switch factor which converts PIP3 back to PIP2 in the pathway, okay? So usually cancer cells often have a lower activation of this P10 molecule. And this molecule often shows STAT3 hyperactivation. So cancer cells deficient of P10 molecule usually show STAT3 hyperactivation, making the dual inhibition a necessity. Okay. So compounds, since currently we are in the first phase of the drug discovery process, we decided to retrieve compounds from available literature. All right. So based on gas chromatography, mass spec, and LCMS data available on in publications, we chose these compounds: alpha-hedrin, stigmasterol, campistrol, beta-sitosterol, and lotinib is a standard. It's a standard oral anti-cancer drug. Okay, next one. Yeah. So, and we have sacituzumab, which is also a standard drug, and we have thymoquinone. This is the most abundant compound found in Nigella sativa, that's the black seeds, okay? So we use that in this study too. Then we have nigelidine and also quercetin. The sources are seen there, that's black seeds, they are all found in black seeds, and even in some plants too, all right? So the methodology of this study followed the usual target and compound retrieval using database from databases such as Zinc database, that's where we got our compounds, and PubChem. Then there is TC.